Welcome to NATA's introduction to ISO 15189 2022 Medical Laboratories Requirements for Quality and Competence. This presentation will focus on the significant changes between the 2012 version and the 2022 versions of the standard. It is not intended to be a presentation on all standard requirements. Laboratories are reminded that NATA has revised and updated its 15189 training to include the 2022 version of the standard and its requirements. This training also incorporates the requirements of NPAC and the TGA as they applied to Australian laboratories. Details of this course provided by NATA Training Services is available on the NATA website. The 2012 version of the ISO standard that we all know and love will no longer be supported, like your favourite operating system or version of software. The 2012 version of the standard has been adopted by over 60 countries as part of their mandatory medical laboratory accreditation requirements, and it was one of the fastest growing international quality standards in the world. But when the standard was introduced, labs were not as automated or interfaced as they are now. There was no COVID. Molecular testing was limited, and next generation sequencing was the new kid on the block. Now molecular testing is mainstream. There is a whole new profession known as bioinformatics, and AI is the new buzz phase. These and other developments limited the applicability of the 2012 version of the standard. ISO 15189 is a global ISO standard. ISO, through its members, brings together experts to share knowledge and develop voluntary, consensus-based, market-relevant international standards that support innovation and provide solutions to global challenges. ISO has over 160 members globally and has published more than 23,000 standards. It has been in operation for more than 75 years. Its guiding principles include developing standards that respond to a need in the market. New work items can be proposed by any member countries and are approved by vote. Standards are based on global expert opinion, with subject matter experts from all interested countries working on documents. ISO standards are developed through a multi-stakeholder process, with experts coming from industry, users, academia, NGOs and government. And ISO standards are based on a consensus, with all comments from stakeholders taken into account. So why and how has the standard changed? As we have seen, there have been significant changes in the laboratory landscape since the 2012 version of the standard was published. In addition to this, all ISO standards are subject to review every 5 years to determine if they remain as is, need to be revised, or need to be withdrawn. ISO 17025 is the normative reference for ISO 15189, and as such, 15189 is required to follow a similar structure and incorporate the same mandatory wording. 17025 was reviewed and updated in 2017, and the 2012 version of 15189 no longer follows the same structure or mandatory wording requirements. Adding to the pressure for change, in 2017, prior to the review, a survey was sent out to users asking members for issues in the application of the standard. There were some 1,600 responses, so a revision was required. A team was assembled which had strong Australian representation, including Associate Professor Beverly Rowbotham, the current chair of MPAC, as chair of one of the working groups, and NATA's own Andy Griffin. ISO operates by consensus ballot. Only when there is a general agreement does the document move to the final stages. Each one of the comments received had to be reviewed and considered. There were thousands of comments received for each level of development. ISO has also introduced a standard structure for its standards and some mandatory wording requirements. These had to be incorporated into the standard. After 5 years, finally, on 4th of December 2022, the new version of the standard was published. The structure is significantly different with the focus on operations, with management system support. The new standard had 3 drafting objectives. There was a greater focus on the patient and patient care. There was a greater focus on the importance of risk management for the laboratory and for the patient. And there was a requirement for a decrease in prescription, allowing more opportunity for labs to do things in a way that meets their needs. The definition of customer has been reviewed, and we are now focusing on the patient. The writing committee was strongly motivated by patient wellbeing and all patients having access to a fair and a just culture in healthcare. In Australia, this is not an obviously apparent concern, but this is a global standard. In other world economies, fair and just access to healthcare can be a real issue. Fair and just is not the norm. Examples include patients that have been discriminated against on the basis of their infectious status, including HIV, or of their race, skin colour, or gender. The focus is on healthcare for all. This has been captured in the standard in clause 4.3, where the laboratory shall establish and implement processes that uphold the rights of patients to care that is free from discrimination. And again in clause 7.7.3, the resolution of complaints, where the investigation and resolution of complaints shall not result in any discriminatory action, such as the refusal of services. With regards to the focus on risk management, the word risk is included in all of these listed clauses and is mentioned more than 80 times. The emphasis is not only on risk, but is risk and opportunity. Due to the less prescriptive nature of the document, labs have the opportunity to address risks in a way that best suits their needs, their resources, and their operating environments. Prescription in a standard refers to a mandatory requirement that must be followed to comply. Prescriptions are usually not negotiable, but must be adhered to for compliance. There was an ongoing objective to reduce prescription in the standard. Labs are still required to have a process, but they are no longer restricted by having to have a procedure in a lot of cases. Labs have the flexibility to find their own solutions, but will need to demonstrate that they not only cover the requirements, but that they consider the associated risks. The term procedure is still in use, so labs will have to review these opportunities carefully. Just a quick note on the distinction between process and procedure. Process is usually general in nature and describes the overall approach. It's the goal or objective the lab sets out to achieve. Procedure is the specific set of instructions for how a task is to be performed, the step-by-step guidance on how to perform that task. In the version of the standard, in some clauses, where previously there was a requirement for a procedure, there is now a requirement for a process. This ties in with the objective of less prescription and opens opportunities for lab to do things differently, considering the risk. To provide some examples where process has replaced procedure, in the 2012 version of the standard, under equipment, the lab shall have a documented procedure for the selection, purchasing and management of the equipment. In the 2022 version of the standard, the lab shall have a process for the selection, procurement and acceptance testing. And another example, in the 2012 version of the standard, identification and control of non-conformities, the lab was required to have a documented procedure to identify and manage the non-conformities. In the 2022 version, the lab shall have a process for when any aspect of lab activities or examination results do not conform with its procedures. And one final example, in the 2012 version of the standard, under the resolution of complaints, the lab is required to have a documented procedure. In the 2022 version of the standard, the lab shall have a process for handling complaints. It should be noted that not all prescription has been removed. The requirement for a procedure is still in the document more than 90 odd times. This slide has been included to highlight the most common non-conformances and risks that NATA identifies when conducting assessments. These include issues with quality control, participation in external quality assurance programs, staff training and competency, validation and verification of procedures, and equipment being fit for purpose. The new version of the standard strengthens the requirement for each of these working areas to reduce risk and increase patient safety. The 2012 version of the standard had 3 simple sections. This has been expanded and enhanced in the new version. There are now 8 sections, and this reflects the structure of other standards developed by ISO. As previously mentioned, the standard emphasis has changed, with the focus now on operations, with management system support. And governance is in the spotlight, with the role of the lab director more clearly defined and highlighted. Looking firstly at sections 1, 2 and 3, which covers the scope, normative references, terms and conditions. The scope of this document specifies requirements for quality and competence in medical laboratories. It now includes requirements for point-of-care testing. The normative references for the document remain unchanged. However, as previously noted, these references have been updated since the previous version of the document. There have been a number of inclusions in the terms and definitions. There is now the patient, and the word customer is no longer used. Also included is internal quality control, in vitro diagnostic medical device, and examination procedure. The definition of process has been excluded. The quality management system, policies and objectives has been replaced by the management system, and the definition of lab director has been included as lab structures were too variable to encompass this definition. Commutability of a reference material, commutability, is a definition that has been added. Commutability, in very simplified terms, refers to how close a reference material is in providing the same or similar results for 2 different methods. The higher the result, the better applicability of the reference material. Patient samples are preferred when comparing different reagent lots to avoid issues with commutability of quality control materials. This could be extended to the preferred use of patient samples when verifying new equipment. This is an issue labs have and a common assessment finding. Section 4 is the general requirements section and covers impartiality, confidentiality, and requirements regarding patients. Lab activities shall be undertaken impartially, free from bias and conflict of interest. The lab shall be structured and managed to safeguard impartiality, and there shall be a commitment to impartiality from management. The lab shall not allow commercial, financial, or other pressures to compromise impartiality. Labs are required to monitor activities and relationships to identify any threats to its impartiality, and this monitoring shall include relationships of its staff. Where threats to impartiality are identified, they shall be eliminated or minimised so that the impartiality is not compromised. Labs shall be able to demonstrate how it mitigates such threats. Some practical examples of how labs can strengthen their impartiality include conflict of interest policies that identify potential conflicts of interests and procedures for managing these conflicts, establishing an impartiality committee to oversee and manage issues, personnel management including the recruitment, training, performance management and code of conduct, and managing relationships with external stakeholders. With impartiality, the objective is to safeguard the validity of results, avoid conflicts of interest, and ensure that the management structure, use of resources, and undertaking of processes are implemented and maintained in a way to avoid any compromising situations or actions. Section 4.2 deals with confidentiality. A breach of confidentiality can lead to loss of customer confidence and loyalty, but it could also result in costly lawsuits. Labs are responsible for the management of all patient information obtained or created during the performance of lab activities. This includes patient privacy and confidentiality. The lab shall inform the user and the patient in advance of information it intends to place in the public domain. When the lab is required by law or contractual arrangements to release confidential information, the patient concerned shall be notified of the information release unless prohibited by law. Information about patients from sources other than the patient, this could include regulators or complainants, shall be kept confidential by the lab. The identity of the source shall also be kept confidential by the lab and not shared with the patient unless agreed. All personnel with access to laboratory information shall keep confidential all information obtained during the performance of lab activities. Some practical examples in a laboratory include the handling of paper documents detailing patient information, secure network storage, confidentiality training, or even mobile phones in a lab setting. The requirements regarding patients is new, but will not be unfamiliar to labs in Australia accredited to the MPAC requirements. This section is similar to the requirements in Section 2 on ethical practice of the RNPS. The focus is firmly on the patient. Labs are required to establish and implement processes ensuring the patient's well-being, safety and rights, including providing opportunities for feedback and informing the patient not only of testing requirements, but turnaround times and any associated testing costs. Labs are required to continually review their test offerings to ensure they are clinically appropriate, necessary and fit for purpose. The lab shall establish and implement a process of disclosure to patients and users of incidents that resulted or could result in patient harm, and the records of actions taken to mitigate those harms. Full open disclosure. Labs shall have a process for the respectful treatment of patient samples and patient remains. Labs need to consider how this extends to culturally sensitive matters, including Indigenous populations. Labs need to ensure informed consent is obtained when required. This particularly applies to molecular testing, where there are not only associated costs, but the possibility of incidental findings. Labs will need to have a process in place that protects patient samples and records, including when there is a lab closure or change of ownership. And labs shall have a process for making information available to the patient or other healthcare providers when requested. Labs shall uphold the rights of the patient free from discrimination. As previously stated, the requirements of this section are similar to the requirements of the local NPAC RNPS document. Labs will need to make very few changes to satisfy this clause. Section 5 covers the structural and governance requirements. The requirement for a laboratory to be a legal entity remains unchanged and now includes a note clarifying that government labs are recognised as entities. Section 5.2 relates to the role of the laboratory director, who has ultimate responsibility for laboratory operations, reaffirming the MPAC role of the designated person. Noting there is no definition of laboratory director due to the variability in lab structures covered by this global standard. The lab shall be directed by persons, however named, with appropriate qualifications, competence, authority and resources to fulfil the requirements of the standard. The lab director responsibilities need to be documented, one of the only documentation requirements in the new standard. These responsibilities include the application of the risk management framework to all aspects of the lab's operations, including sample collection, sample transport and data entry that we sometimes see is not considered as part of the director's role when NATA undertakes its governance assessments. Through the risk management process, risk to patient care and opportunities to improve are systematically identified and addressed. The director may delegate duties and responsibility to qualified and competent persons, and if such delegation occurs, it needs to be clearly documented. Notwithstanding the delegation, the director still maintains the ultimate responsibility for the overall operation of the laboratory. Section 5.3 relates to laboratories' activities. Labs are required to detail and document the range of activities for which it conforms with this document. This includes activities performed at sites other than the main location, for example, point of care testing and sample collection. For NATA, this will be reflected in the laboratory's scope of accreditation. The lab is not directly responsible for testing undertaken in referral labs and cannot claim conformity for tests sent out on a regular basis. As previously required, labs need to ensure advice and interpretation are available to meet the needs of the patient and users. The standard now includes for point of care testing. Under Section 5.4, Structure and Authority, the lab shall define its organisation and management structure and its place in any parent organisation. This will be covered by an organisation chart showing the responsibilities and lines of communication of all staff affecting the results of lab activities. NATA has previously reviewed all charts on assessment, but the lab's place in the parent organisation is something that may need to be addressed by some of the pathology networks and even the hospital networks. What is the facility's place in the parent organisation? The lab shall specify procedures to the extent necessary to ensure consistent application. It is up to the lab to determine the extent necessary. NATA will review with interest how labs have determined what extent is necessary for consistent application and valid results. The new standard expands on the roles and responsibilities for quality management, affirming that there shall be personnel responsible for the implementation, maintenance and improvements of the management system who shall have the authority and resources needed. Noting also there is no longer a requirement for a specific quality manager. However, a quality manager is still an MPAC RMPS requirement. Under Section 5.5, lab management shall establish objectives and policies that are measurable. Quality indicators established to evaluate performance throughout key aspects of pre-examination, examination and post-examination processes need to be linked to management objectives and need to be consistent with policies. For example, to be meaningful, they need to be linked to audit results or management review and objectives, not just a list of turnaround time, sample rejection rates, etc. Section 5.6 deals with risk management, which is something labs in Australia are very familiar with and which has guided accreditation activities for some time now. Labs shall establish processes for identifying risks to patients and opportunities for improved patient care. The lab shall develop actions to address both risks and any opportunities for improvements identified. And the lab directors shall ensure that these processes are evaluated to determine how effective they are, and if ineffective, the processes shall be modified. This review process is reflected in MPAC RMPS and acts as a framework for the prioritisation and allocation of resources. Section 6 of the standard deals with resource requirements. Labs shall have available the personnel, facilities, equipment, reagents, consumables, and support services necessary to manage and perform its activities. Labs shall have access to enough competent staff to perform its activities impartially, ethically, and in accordance with the lab's management system, previously the quality system. The competence requirements for each lab activity that influences patients' results, including education, qualifications, training, and technical knowledge, shall be specified, and the lab shall ensure all personnel have the competencies to perform the activities for which they are responsible. The lab shall manage the competence of its staff. This includes the requirements for the frequency of competency assessments, and shall have documented information demonstrating competence of personnel. There is a greater focus on the authorisation of staff to perform specific lab activities, including the selection, development, modification, validation and verification of methods. Staff also need to be authorised for the reporting of results and for the approved use of lab information systems, including to change patient data or examination results. Personnel records still need to be maintained and include competence monitoring and authorisation. The requirements for facilities and environmental conditions necessary for the performance of lab activities shall be specified, monitored, and recorded. This has become more critical as lab equipment has become more automated and precise. The environmental conditions shall not adversely affect result validity. This includes sites other than the main lab where examinations are performed, including point-of-care testing. Facility controls include control of access, prevention of contamination or cross-contamination by lack of separation. Storage that ensures continuing sample integrity shall be provided and samples stored to prevent cross-contamination deterioration. Hazardous materials shall be dealt with appropriately. Personnel facilities include adequate access to toilet facilities and a supply of drinking water, meaningful requirements in less developed economies. Under Section 6.4, labs shall have access to equipment required for the correct performance of activities. There shall be processes for the selection, procurement, installation, acceptance testing, maintenance, and decommissioning of equipment in order to ensure proper functioning and to prevent contamination or deterioration. Equipment shall be uniquely labelled or identified, and an equipment register shall be made, which was not a previous requirement. Of note, equipment includes the hardware and the software, the measuring systems and the lab information systems, and the sample transport systems, including hospital pneumatics and lines. Labs shall verify that the equipment conforms to specific acceptability criteria before being introduced or even returned to service. There is an emphasis on equipment used for measurement. It shall be capable of achieving the accuracy or uncertainty required to provide a valid result. Equipment shall still be safeguarded to prevent unintended adjustments, shall be operated by trained and competent staff, shall have instructions for use available, and shall be used as specified by the manufacturer unless validated by the lab. Labs shall have preventative maintenance programs based on the manufacturer's instructions, and any deviations from schedules or instructions shall be recorded. Records for each equipment item that influences results shall be maintained and be available for at least the lifespan of the equipment, potentially longer. Metrological traceability is required for both quantitative and qualitative methods. Labs shall have procedures for the calibration of equipment that affects exam results, including where correction factors are used, how these are recorded, and how to handle cases where calibration is out of control to minimise patient risk. Labs shall establish and maintain metrological traceability by means of a documented, unbroken chain of calibrations and ensure that results are traceable to the highest possible level of traceability. For genetic examinations, traceability to genetic reference sequences shall be established. This is a new requirement that will need to be worked through. Qualitative and quantitative methods that measure characteristics rather than discrete analytes shall specify the characteristic being addressed and the requirements for reproducibility of these over time. Under Section 6.6, labs shall have a process for the selection, procurement, reception, storage, acceptance testing, and inventory management of reagents and consumables. They shall be stored according to manufacturer's specifications and conditions monitored where relevant. This includes goods that are received and stored off-site. Each reagent or new formulation of reagent, new lots or shipments shall be verified before placing into use or before results are released. Patient samples are preferred when comparing different reagent lots to avoid issues with commutability associated with QC materials. Instructions for use shall be available for reagents, and they shall be used in accordance with manufacturer's instructions. Records for reagents shall be maintained, including the date of receipt and conditions when received, the expiry date, the date of first use, which was previously the date of entering into service, and, where applicable, the date the reagent or consumable was taken out of service. In-house reagents need to include reference to the preparer. With regards to service agreements, Section 6.7, labs shall have a procedure to review service agreements, and users shall be informed of any changes that can affect patients' results. Potentially, this includes a change in platform or matrix. Labs shall inform users of activities performed by referral labs and consultants. And where labs support point-of-care testing in other parts of the organisation, there shall be service agreements to ensure respective responsibilities and authorisations are detailed and communicated. Section 6.8 deals with externally provided products and services. Labs shall ensure that external products or services are suitable for incorporation into the lab's activities. With referral labs, the referring lab shall communicate requirements and is responsible for ensuring examination results are provided to appropriate persons. The lab shall maintain a list of referral labs and consultants and have procedures for the referral of samples and evaluation of external providers. Section 7 deals with process requirements. Process requirements consider the best interest of the patient. The lab shall identify potential risks to patient care in the pre-examination, examination, and post-examination processes. Risks shall be mitigated to the extent possible, and mitigation efforts shall be monitored and evaluated. The lab shall also identify opportunities to improve patient care and develop a framework to manage these opportunities. There shall be procedures for all pre-examination activities. Information available to users and patients, including requirements for consent and the lab's complaint process. The lab shall have a procedure for managing oral requests that includes documented confirmation within a given time. Lab shall have a procedure for the collection and handling of primary samples, and deviations from the established collection procedures shall be clearly recorded. It shall provide instructions for pre-collection activities, such that sample integrity is not compromised. This shall include instructions on the order of sample collection to prevent carryover. This also includes special collection timing where relevant and sample labelling for unequivocal patient identification. Patient consent is important, especially for special procedures with an increased risk of complications that may lead to more detailed explanation and, in some cases, record consent. Collection activities include verification of the patient and recording the identity of the patient collecting the sample along with the collection date and time when relevant. Labs shall provide instructions for the packaging and transport of samples to ensure ongoing integrity. It shall periodically evaluate the adequacy of the sample transport system. Samples shall be unequivocally traceable to patients at all times, and when applicable, this includes the anatomical site. The lab shall have well-established criteria for acceptance and rejection related to patient risk, and there shall be procedures for handling urgent samples. Labs shall have a process that considers the best interest of the patient when a sample has been compromised. When a compromised, clinically critical or irreplaceable sample is accepted, after consideration of the risk to patient safety, the final report shall indicate the nature of the problem and, where applicable, advising caution when interpreting results that can be affected. Labs shall have procedures for appropriate storage, and where additional testing is required on the sample, there will be time limits on request. The time between sample collection and testing shall be specified and monitored where relevant. Labs shall select and use examination methods which have been validated for their intended use. All procedures, including instructions, manuals, and reference data, shall be kept up to date and available. Staff shall follow established procedures, including point-of-care testing operators, and authorised staff shall periodically evaluate methods provided to ensure they are clinically appropriate for the requests received. Labs shall verify procedures, ensuring the manufacturer's required performance can be achieved before introduction to the extent necessary to ensure valid results. Staff with appropriate authorization and competence shall review verifications, and records shall include a statement of whether the performance specifications were achieved, and if not, what action was taken. Labs shall validate developed methods or methods used outside their original intended purpose. The validation shall be as extensive as is necessary and confirm that the requirements for the intended use of the examination have been fulfilled. Staff with the appropriate authorization and competence shall review validations, and records shall indicate a statement on the validity of the method detailing its fitness for intended purpose. Biological reference intervals and clinical decision limits shall be defined to reflect the patient population while considering risk to patients. They shall be periodically reviewed and changes communicated when applicable. Labs shall document procedures to the extent necessary to ensure the consistent application of activities and the validity of results. If abbreviated documents are used, they shall correspond to the procedure, and all documents associated with processes shall be document controlled. Labs shall have a procedure for monitoring the validity of results. Data from monitoring activities shall be recorded in such a way that trends and shifts are detectable. Statistical analysis shall be applied to review results. Quality control material shall be appropriate to clinical decision making, and third-party material should be considered for ongoing monitoring. The material should remain fit for purpose and have a matrix as close as possible to that of the original patient samples. If QC material is not available, labs should consider using other methods, including retained patient samples. QC frequency should be based on the stability and robustness of the examination method and the risk of harm to the patient from an incorrect result. QC data should be reviewed at regular intervals that allows for a meaningful identification of current performance. In the event of QC failure, the lab shall prevent the release of patient results. Labs shall monitor performance by comparison with other labs. This includes participation in external quality assessment programs, including for point-of-care testing. These programs shall include samples that mimic patient samples and meet target values. Alternative comparison methods shall be used when available EQA programs are not available, with labs justifying the alternative choice. Again, data shall be reviewed regularly to allow for meaningful identification of current performance. Results shall be reported accurately, clearly, and unambiguously. Reports shall include all available information necessary for the interpretation of the results. If results are delayed, the lab shall notify users based on patient risk. Responsibilities and procedures for how results are released for reporting, including by whom and to whom, shall be specified. When exam results fall within specified, established critical decision limits, authorised persons shall be notified as soon as relevant. This needs to be carefully documented, including details of the person notified. There needs to be an escalation procedure when a responsible person cannot be notified. These requirements are reflected in the impact requirements for high-risk reporting. Note that when agreed with the user, the results may be reported in a simplified way, with any information not reported being readily available. Where results could potentially require special counselling, some genetic reports, or infectious diseases, labs should ensure that these results are not communicated to the patient without the opportunity for adequate counselling. For automated reporting, there shall be a procedure detailing the criteria for release, and where necessary, there shall be an ability for rapid suspension. The standard includes detailed requirements for inclusions in reports. Unless the lab has documented reasons for omitting any items, this will help labs keep up with requirements as systems change. Report interpretation comments shall include comments on discrepancies when testing is performed by different procedures, such as point of care testing or in different locations. When there are amendments to reports, the emphasis is on traceability. Section 7.5 deals with non-conforming work, and the lab shall have a process for when activities do not conform to its own procedures. This includes evaluating the clinical significance of the non-conformance, and actions taken shall be commensurate with the risk to the patient and of a reoccurrence. Under Section 7.6, labs shall have access to the data and information needed to perform lab activities. The lab is ultimately responsible for the lab information systems. Information systems shall be validated and verified by the lab before introduction, and any changes, including upgrades, shall be documented and verified. Systems shall be implemented taking cybersecurity into account to protect from unauthorized access and safeguard data against tampering or loss, and shall be maintained to ensure data integrity. The NPAC Information Communication Requirements document extensively covers off on these requirements. Labs shall have processes in place to maintain operations during system downtimes. This includes automated reporting of results. Labs shall have a process for handling complaints that includes the process for receiving, investigating, and resolving the complaint. A description of the process for handling complaints shall be publicly available. The resolution of complaints shall be reviewed and approved by persons not involved in the subject of the complaint. Where resources do not permit this, an alternative approach shall not compromise lab impartiality. Section 7.8 is new and deals with continuity and emergency preparedness planning. Labs shall ensure that risk associated with emergency situations or other conditions where lab activities are limited or unavailable have been identified, and that there is a coordinated strategy that involves plans, procedures, and technical measures to enable continued operations. This includes such things as flood, fire, earthquake, and storms. Plans shall be periodically tested and the planned response capability exercised where possible. Plans need to consider staff capabilities, the training requirements, and proportional actions appropriate to the magnitude of the emergency and its potential impact. And finally, Section 8 deals with management system requirements. Labs shall establish, document, implement, and maintain a management system to support the requirements of this document. At a minimum, the management system will include responsibilities, objectives and policies, actions taken to address risk and opportunities, continual improvement, and management review. A facility seeking accreditation to ISO 15189 may establish a management system in accordance with ISO 9001. In such a case, the system may not be assessed in full by NATA if the following is in place. The management system is certified by a recognized accredited certification body, such as JASANS, which is accredited to certify management systems. NATA will request the facility to provide evidence of the certification body's scope of accreditation. Copies of the lab's most recent certification audit reports will need to be made available for NATA for review, including confirmation of the closeout of any nonconformities. There will need to be evidence that the management system supports and demonstrates the consistent fulfillment of the requirements of 15189 for the activities covered by the NATA scope of accreditation. The required extent of assessment will then depend on the detailed evidence provided. If NATA identify any nonconformities with the management system, they will be reported under the relevant Section 8 clauses. Where the lab's management system certification status changes, the facility shall notify NATA within 14 days. If the lab has adopted a management system that has not been certified by a recognised certification body, the NATA will assess the management system in full against the clauses of Section 8. This was a new option in 17025 when it was released in 2017, and approximately 50% of labs adopted the use of the certified management system. Other management system requirement changes include the standard no longer prescribes a quality policy or specifically a quality manual. With regards to the control of records, the prescriptive list of what a record is no longer included. The retention of records is to be based on risk. Locally, this is determined by impact. Labs shall identify risks and opportunities for improvement and shall prioritise these based on risk. Actions taken shall be proportional. Continuous improvement activities shall be directed at areas of highest priority based on risk and opportunity. Impact of any nonconformities in relation to risks and opportunities must be determined. Evaluation and audits to demonstrate that the needs of the patient and the lab user have been met and any risks to patients identified. And the management review input needs to include point of care activities. Annex A is new and details the additional requirements for point of care testing. The organisation's governing body is ultimately responsible for ensuring that appropriate processes are in place to monitor the accuracy and quality of point of care testing. Labs shall appoint a person with appropriate training and experience to be responsible for point of care testing quality. And labs shall appoint a person with appropriate experience to manage training and competency assessments of personnel performing point of care testing. The trainer shall develop, implement, and maintain an appropriate theoretical and practical training program for all personnel. NATA will start assessing to 15189 2022 from the 1st of August. As I hope I have demonstrated, the revision of the standard will not require labs to extensively rework their management systems, but it will allow for greater flexibility and a greater focus on patients. NATA will be holding 2 Zoom sessions to address any questions that you may still have after watching this information session. These sessions are open to all human pathology authorised representatives and technical assessors. The sessions will be held on the 4th and the 6th of July, and details can be found on the NATA website. Thank you.